So what I'm trying to do here is just go through the fundamentals. Like what are biofilms? How do they develop? How do they behave? And how do we study them? And then something about their impact. So if we look at this, the way we look at biofilm, these are microorganisms that colonize the surface. That's by definition a biofilm. So then what we see here, we all hear about and see suspended microorganisms, in this case swimming around in some water, but then when there is a surface, they tend to colonize the surface, and when organisms have colonized, they will start growing on this surface. First in a very thin layer, then thicker layers. It can be smooth, or it can be like very irregular colonies, like shown in this illustration. And then at some point, we will also have microorganisms leaving the biofilm and going back into suspension. Some of that will happen all the time, and then this illustrates a specific episode that we will get back to. I also want to point out that these slides are from Center for Biofilm Engineering at Montana State University in the US, where I was a student many years ago. So pretty much everything in this presentation comes from there, partly mine and partly from others. So one thing that generally we look at biofilm as these organisms on the surface, and they get the food from the liquid. And that's how, for instance, we treat wastewater. Pollution in the liquid diffuses into the biofilm and is being consumed or converted, and then we have a treatment process. But what I will be showing here is that biofilms has a much wider relevance. Biofilms are pretty much everywhere. So we'll get back to that also a bit more. But one of the things that I'm starting out with here is that often the microorganisms can also consume the surface they're growing on, even if it's not organic. For instance, here we have an electrochemical reaction where the organisms are actually consuming iron. So they're eating iron. This was a pretty controversial topic way back when, even when I was a student. I remember my advisor, who was kind of Mr. Biofilm, he was trying to convince the industry of this being a big problem. Now it's well recognized as being a big problem. I've seen somewhere it's been claimed that, for instance, in the offshore installations in the North Sea, corrosion caused by microorganisms is a very big technical and economical problem. So this is now well recognized. But what is interesting here is that we can also take advantage of this electrochemical reactions. We can, for instance, deliver electrons to the microorganisms growing on such surfaces. So we can have anodes and cathodes with microorganisms that we can put into our bioreactors and make them do things more efficiently. So we have just recently received a large grant to study that. So maybe some of you will have an opportunity to go deeper into that as part of your master thesis. We are presently hiring postdoc and PhD students for this new project. And the application for us in this case is to put electrodes into anaerobic digesters to make them more efficient. So you know that when we run our biogas process, we produce methane and CO2. What we are aiming at is to also convert that CO2 into methane by supplying electrons, by supplying protons through electrodes. So this is could become a very important future application of what we then call bioelectrochemical reactions. It's a fairly new research field. I think the first article where somebody actually used microorganisms growing on electrodes to produce electricity was around the year 2000. So that's another way you can do it. You can make the organisms by consuming and working on mineral or metal surfaces. They can actually set up a current. But what we're doing is trying to do the opposite, supplying a current to make them do something else with the wastewater that we're treating, to make that more efficient. We also think there are many other applications than CO2 to methane. It can be ways to enhance degradation of difficult chemicals. So there are many possibilities and options. But it's a new field, so it's not something where we're moving along really fast. And as I said, and what we're trying to do is try to utilize these phenomena in a positive way. But I see now that the Center for Biofilm Engineering, when that was established as a national US center for research on biofilms, it was not so much as a method to treat water or wastewater, but rather because biofilms cause all kinds of problems. So, and today also, most of the funding comes from mainly two areas where biofilms represents a problem. And the first area is industry and similar cases. For instance, cooling water in industries can, where you have heat exchangers, if you get biofilms on the heat exchangers, you get fouling. You've learned about this in other courses. And these biofilms can then lead to reduced capacity. And I remember at the center, when I was a student there, we actually got funding from the nuclear power industry to find better ways to control this biofilm. Partly because they were forced to not use so much chlorine, because they were polluting the surroundings when they were loading their all heat exchange and everything with high amounts of chlorine. So they wanted more environmental friendly ways to control the biofilms. Then certainly we have food processing industries, and even in the barn where you have cows, milk machines, biofilms used to be a big problem. They would grow inside the pipes and the pumps and the machines and then contaminate the milk, and it would go bad fast. So in that case, there are a few different ways of dealing with it, but the standard way in Norway today is that they milk the cows twice a day. So then every morning they will flush the system. After the milking, they will flush the system with a strong acid. Now, there are microorganisms that can live very well with strong acid. But then, after the milking in the evening, they flush it with a strong base. And there are no microorganisms that can handle both strong acid and strong base. So by alternating every day between acid and base, they keep their system safe. Actually, we'll get back to that a little bit next time when we talk about treatment of small decentralized systems, because that often includes that type of wastewater. So if we kill all the microorganisms to avoid biofilms, we may also have trouble treating that water afterwards. But there are quite simple solutions to that. Oil and gas, industry, biofilms, big problem. I already mentioned how it's a big cost for the oil industry doing offshore operations, especially the biocorrosion, but also even inside oil reservoirs you get biofilms, and they can cause problems. You've learned about hydrogen sulfide in this course, and one of the big problems is hydrogen sulfide development in oil reservoirs. And then what you get is what is called a sour oil, which has much lower value than a good quality oil. And since they use in the oil reservoirs, especially offshore, they pump seawater in there to maintain the pressure. So they have wells where they pump water in and then they push oil out in other wells. But if you at the same time push in sulfur, which there is plenty of in seawater, and there are microorganisms, all of a sudden you have an environment where you produce hydrogen sulfide. Serious and very expensive problem to deal with. Ship hulls, we get biofilms growing on boats. This is illustrated here by a naval ship, and that is for a reason, because part of the funding at this Center for Biofilm Engineering was from NATO, because this how to keep less growth on the ships so that the ships can move fast in the sea. Because what happens is when you have a biofilm on a ship hull, the ship goes slower. It adds friction. And for anybody who's into sailing, they know this for sure. So during the season, when you sail regatta, you have to clean the ship hull a few times to just be able to compete. Otherwise, it gets slower and slower. So that, and in this case, the methods available are these paints with some type of toxic chemical to keep the microorganisms away. Copper-based is the most popular, but again, restrictions on how much you can put on and causing local pollution and such makes it difficult to keep the ship hull clean and complying with environmental regulations. So those are what we can say sort of industrial problems caused by biofilms. Well, also paper mills have big issues with this, but in general have figured good ways to deal with it. But when they don't have it, it can reduce the quality of the paper produced, for instance, if you also have living microorganisms mixing in with your blend. Then we have drinking water. We have talked about drinking water. We've been to the drinking water plant, and one of the issues there was these barriers for contamination, microbial contamination. And in Norwegian, European regulation, you need minimum 2 absolute barriers, so that if one fails, the other one is still there, and you always have safe drinking water. And you've seen the plant, and one of the methods they use is UV radiation. So a nice clean water radiated by UV will pretty much kill everything. But it will not keep microorganisms from growing in the pipes downstream, because it only treats the water when it passes the UV radiation. So therefore they also add chlorine that follows the water. So that's an additional barrier at the same time as it is supposed to help avoid growth of biofilms inside the pipes. Now that will generally not be perfectly okay. I mean, there will in general be some biofilms in the pipes. So to avoid then getting microorganisms into your tap water, which can be pretty, it's not necessarily dangerous, because these organisms that are growing inside your pipes may not be harmful, but it may not look good because you may have microorganisms there. And when you have microorganisms, you also have small animals that feed off of these. And if it's not done properly, all of a sudden you see little organisms moving around in the water you're going to drink, and you don't want it. Right? So that's another challenge. And one of the cases where you don't want to dose your chlorine so high that it stinks when it gets to your use. So in general, there is a trade-off. You accept that there is a little bit of microbial activity in your pipes, but not so much that it's ever dangerous or visible. Then we have our teeth. I assume we all brush our teeth. Why? Well, to limit the biofilm. Really. And if we think about what happens when we get trouble with our teeth, it's really similar to what we have here. Consider this to be your teeth, and you have microorganisms growing on them, and all of a sudden you get the electrochemical reactions and tearing away on the teeth, and you get holes going in, and you need to go to the dentist. But it's pretty much the same phenomenon. Often you have some aerobic zones, and if you don't clean your teeth properly, you get some zones that are anaerobic, and then you get currents moving, and these are issues that we deal with simply brushing our teeth. If we go to the next here, the last one, medical implants. That has turned out to be a really big area. Lots of funding coming from there for the biofilm research. If it turns out more and more people, when they get old, they replace parts of their body. Here shown probably a hip joint. And if that's not perfectly sterile, you may start getting a biofilm where you deal with this, and then you're in trouble. And it may have to be redone. Another issue is that more and more of these microorganisms that you find in hospitals are resistant to antibiotics. So you may get them in there, and then the attempt to cure is to give you antibiotics, and they may not work. And this is a growing concern, for sure. So let's take a look a little bit more at the body and sites of biofilm infections. We already mentioned the teeth, but also the rest of the mouth. Here, hip implant that we just talked about. It can also be when you're in the hospital and they need to supply nutrients or drugs into your veins, and that is another serious case where you can get infections. I also just yesterday I came across an article in Science, just fresh article about how you can also have trouble with in your intestines, your stomach, colon, where we really have lots of microorganisms. We depend on having lots of microorganisms. In general, we have more microorganisms than human cells, each one of us. So we are just carriers, we're just these very advanced support systems for microorganisms. We feed them, we take care of them, but this article shows that some of the things that are put into foods these days, preservatives, for instance, can alter the ability of the microorganisms to grow, so that we are causing a shift in the microbial flora inside our body that is not good. So this is actually, according to that article, it's almost like an epidemic. People are getting sick because of this. And the new organisms then colonizing, because typically what happens is that some of the bad organisms that can cause damage to us, they have the ability to break down some of these additives, food preservation chemicals, for instance. Also, it's said, not with just as much certainty, but for instance, these no sugar drinks, we're convinced that we should switch from consuming lots of sugar, so we buy the zero sugar Coke, and that is the replacement, because it tastes just as sweet. And the idea is that the sugar that it has been replaced with just goes straight through our body. But it turns out now that there are microorganisms that are starting to also consume those that can cause health problems. So there is no easy fix to this. But essentially, at least my conclusion from reading that article is that it's better to cook your food from the bottom and not buy too much preserved and very processed food. Okay, so we see that biofilms in the body can be a blessing. We're totally dependent on it, but it can also be a problem when it gets out of control. One more thing. If we have a cold, what is a cold? Having a cold? Well, typically it is a biofilm in our lungs getting out of control. So what we should try to consider is that we should try to control it. And one of the things that I think is often causing us to get a cold is when we're in a stressful situation. There's all this focus on getting infected by people having a cold, but I think the main reason is that our immune system and our body is, for some reason, at the, it's not strong enough. So we're vulnerable to microorganisms taking over. And often, for instance, when we're stressed, we have trouble keeping the body's water level high enough. Right? And what happens then? Well, then the membranes in our mouth and lungs becomes a little wrinkle. And then the microorganisms have very nice sites where they can colonize. and take control. So if you see signs of getting a cold, try to relax, drink water, tea, whatever it takes to just stay comfortable and healthy, and you fight the cold back. So if you should try it, it works. Control your biofilm. Be nice to your friendly biofilm by feeding it good food, and when you see unwanted biofilms developing, just try to fight it back by natural means, just by trying to be strong and overcome it. Okay, that was a little bit on the side of this. So then let's go into the details of the biofilm. And now we're in my own PhD work. This is a drawing I made for my dissertation, where I called schematic biofilm model. Remember how we've talked about we have different kinds of models? We've worked a lot on mathematical models. This is more what we can call a conceptual model. So we have some surface where the microorganisms can colonize and grow, called substratum. Then we get the biofilm developing, and at some point this is then made a sketch of what we can call a steady state biofilm. We've reached a situation where the whatever pollution or organics in the liquid can move in by diffusion into the biofilm. The microorganisms getting the food will grow and keep pushing out, and then it's controlled by detachment. So when we reach steady state, we have a balance between growth and detachment. Then it will remain more or less constant thickness, here denoted as LF. But we can also look at it at being different types of biofilm. Typically, most of the detachment will happen out here at the surface, while the growth, maybe also most of it will happen here because they get the food first, and less as we move in. What we also see is that when biofilms become thick enough, the organisms at the surface will run out of food, and we may actually get a detachment happening for the whole film. The whole thing comes off, and it's called sloughing in the book. A common problem in some wastewater treatment plant concepts. In the book, they write a lot about what is called trickling filters. I also talked about it in that other video, so you can learn about that. That's kind of the original way of growing biofilms for water treatment. And in those filters, there is very little shear stress. So the biofilm essentially just keeps getting thicker and thicker. And eventually then you have some type of catastrophic event. It can be that you get this sloughing event all the way from the bottom, releasing large sections of biofilm, not small pieces like here, but large sections. And these sections can then clog the filter, and you have an accelerating problem. And that's why in more modern biofilm reactors, like the one we have at the pilot plant down at the wastewater treatment plant, we have these small plastic carriers, and they move around a lot in the liquid. They collide, so biofilms will kind of, just by collision, that limits their growth. But also we have strong aeration, and these air bubbles cause a strong shear force on the biofilm, keeping it thin and efficient. Then I also made a physical model, a way to study the biofilm on a more fundamental level. Yeah. The whole question, actually, let's go back here. The question my advisor gave me was to find the relationship between detachment rate and shear stress. That was my task. Now, just to let you know already, I never found such a relationship what I concluded was there is no such relationship. And that took some time before it was accepted, but it's now considered reasonable to assume there is no direct relationship. But to some extent, like I just said, without shear stress, the biofilm will just get thicker and thicker. So there is some relationship, but there's not a direct something you can make an equation saying that if you have so much shear stress, you get so much detachment. That is not possible. And the experimental rig I designed for this was made from glass tubes. A recycle pump, feed in, pumping through, and just in a loop and with an effluent. And by having it this way, I could regulate the recycle pump rate, and the shear then on the biofilm inside the tubes would be directly proportional to the pump rate. So what I tested was pumping at different rates, turning the flow up and down and looking at how it influenced detachment. Also, you see here some manometer just to see when the biofilm grows, it will cause friction. So indirectly, by looking at this, I could say something about biofilm development, but even more so, I could say something about the shear, because the shear rate is proportional to the pressure difference that we can measure this way. Also, you see points 1, 2, 3, 4, 5, 6, 7, 8. All these points I would send a light beam through to measure, let's call it just turbidity. So the more biofilm, the less light would go through. So I could then measure pretty much directly how much biofilm was here at any given time. So I started with a clean system, just added an inoculum and watched the biofilm develop, and then see how it changed when the pump rate changed. And later, afterwards, I did quite a lot of work to correlate this optical method to the actual biomass that was there, and it correlated very well. Also, it was made in such a way, this sits vertically, straight up, so that gravity would not influence how the biofilm, it wouldn't be like microorganisms settling on one surface and not on the other. But I also used the microscope, so every day it was hinged down here, so every day I would kind of just bend it down on the microscope, and then I could measure by looking through here, I could measure the biofilm thickness. And I will illustrate that. So this was a square glass tube, so it was easy to use these optical methods. And also inside the main tube, I put an other tube to have more surface area. And that looked like this. If we look at the cross section, this is the main tube. And then inside there is a smaller rectangular tube just to have more surface area. So now there are biofilm, liquid, biofilm, just empty space with glass, more biofilm, liquid, and biofilm. And when I used the microscope to measure thickness, I would measure it as these regular intervals, so that I not only got kind of an average biofilm thickness, but also I could say something about the shape. Was it very smooth like this drawing suggests, or more irregular? So if we look at a typical experiment, this was, I believe, the first experiment. I started with a certain shear stress and kept it that way till I had a significant biofilm. Then I increased the flow rate, so I increased the shear rate by 50%. Kept it there for a while, then I decreased it by twice as much, kept it there for a while, increased again, back to where I started. So that gave me all kinds of opportunity to look at both level of shear stress, but also the effect of the changes in shear stress. And then I did this with different shears, but typically in this range of numbers, because that's where the experimental setup would allow. And as I said, I measured biofilm mass by this optical density at 8 different spots, locations 1 to 8. And here we see how that developed, starting with a clean surface, very little biofilm, but with time there was more and more biofilm coming, and then at the end it would kind of level off. There wasn't space for any more biofilm, essentially. Question?
If the shear stress is high, then it will increase the detection rate.
That was the idea. That was the idea, but I didn't really see that. And now let's look at, you see here, if I got much higher shear, much higher detachment at higher shear, I should see that here. Because again, if we see when did that happen, at 120 hours, let's see at 120 hours. Do I see a major change? I see a small change, but no major change. And if you look at the slope of the growth of the biofilm, no change. So there was kind of an immediate effect of increasing the shear, but no long-term effect. So there was no direct correlation between shear and detachment. That was the main conclusion, really. But we can look at more data. So now the next figure, I take the average of all of these 8. But by the way, you can also see that the highest growth is in location 1 and 5. The lowest growth is in location 8. So there is a little bit of effect that the farther you get away from the feeding point, the less the biofilm grows. Makes sense. And closest to the feeding point, you get more biofilm. But also after the liquid passes the top of the tube and comes back down, where it's kind of being remixed, it also gets pretty good growth. Not sure how to interpret that. But now let's take a look. Let's take the average of each of these data points. And that's what we see in those squares in this graph. And here you can see it almost more clearly that, yeah, there is no direct or strong effect of the shear stress. But in this graph, I also added biofilm thickness. And this was another big surprise, really, that after about just 1 day, there was it was thick. It was as thick as it was going to get. So the biofilm quickly developed to kind of its natural thickness or something, if you can talk about that. What I did not tell you is that this biofilm was a single cell type biofilm. It was only one microorganism that was multiplied, a Pseudomonas aeruginosa, which is actually a dangerous organism. So it was a bit scary when I cut myself or something. The reason for using it is because it's a health problem. It is also one that we know a lot about because it's been studied very well. It's a well-defined organism. But you see, strangely enough, after about 1 day, it reached 35 micrometer thickness, and it pretty much remained that way. A slight drop even later. But I'll talk a little bit more about this drop, because it's not really that it's getting thinner on the surface. It just kind of, but it starts developing some kind of valleys. And because of that, the average drops a little bit. So that was certainly a surprise and rather strange result for me and my advisor, and hard to figure out. But after doing this with more experiments and seeing that this was the way it happened every time. Just had to believe this is how they behave. So what we see here then, okay, if we go to the next one, we can look at the same data. And we see here the biofilm mass going up, and it's reaching kind of a maximum. You can hardly pack your biofilm denser than this. A little bit more than 50%. There has to be some void between cells. If you take spheres and you try to pack them, there will always be some void in between. And so we see that at the end, the void, the red, was actually less than what was filled by microorganisms. But since it filled up already from the start, there must be just cells very spread out with polymers connecting them. And here we see what it looked like when I did transmission electron microscopy on the same biofilm. Not going through the details how I managed to do it, but you can see very densely packed microorganisms. So the optical results and this graph makes sense. We got a very high density where about half of the biofilm volume was filled by cells. And then in here was room for diffusion of liquid. And what we say then is we have three phases. First, the growth control, then some detachment control, and then steady state where growth and detachment are equal. I think that is if we go a little bit further trying to understand these mechanisms of accumulation and shear, we here see a different way of looking at the data. Here I put together two experiments, number 2 and 3 in my series, and these arrows show the changes in shear. So in number 2, the first change was decrease. In number 3, the first change was increase. And also the size of the arrow indicates the strength of the change. So we see that experiments 2 and 3 were exactly opposite. Right? And still then, when we look at specific biofilm accumulation rate, it is hard to distinguish the two at all. So this shows very clearly that you cannot say there is a direct correlation between shear and biofilm accumulation. And since biofilm accumulation depends on detachment, then there is no such direct relationship. We can see, however, for instance here, in the case of number 2, when number 2 had a strong increase in shear, we had a temporary strong change in cell accumulation. So a strong change in shear did cause some portion of the biofilm near the surface to be detached. So it could be that they're just parts that would have detached within the next day, all of a sudden detaches in just a few minutes because of the change in shear. And here we see the other way, when number 3 has an increase, we also have an increase in, or decrease in cell accumulation. So when cell accumulation drops, it means biomass is lost. But we also see that immediately afterwards, it's compensated for by probably increased growth. So there is, as I said, no direct way of modeling biofilm detachment based on shear. And later, for instance, at the same time as this was going on, Aquasim, our modeling tool, was being developed, focusing on biofilm modeling. And I remember we had long discussions. How could this work with the model? Because it just didn't fit with the old ideas that there was a direct link between shear. But then after they got deep into the mathematics of the biofilm, they said, well, you're right, because it cannot really be directly linked, because then the equations don't work anymore. So that was nice to know. But it took some time and lots of considerations before we got to that level. Now, let's take the same figure, but this time with the logarithmic scale here, which means the negative values will disappear, but all the others should be there. So then it looks like this. And what we see is the logarithmic of specific biofilm cell accumulation. It keeps just dropping with time, less and less accumulation as the biofilm gets full. We saw the pictures before. There's just not room for more biofilm. So then accumulation essentially stops. And again, there is no statistically significant difference between the two cases with directly opposite history when it comes to shear. Now let's just jump away from that. I mean, that was pretty solid conclusion. So there was not that much to do, except I will get back to one more slide at the end. But let's talk a little bit about biofilms and antimicrobial control. Remember how we talked about the microorganisms becoming resistant to antibiotics? This is a serious problem, and this graph was made partly to explain that. Here we see a biofilm, and out here, let's say this is some hospital equipment, there is a biofilm, and they add some what's it called again? Antimicrobial agent of some sort. Penicillin was the original one, right? But there are so many others now, but more and more are becoming useless because of this resistance. And here what happens, and part of the explanation why it has developed so fast, is that people didn't really know about the mechanisms of biofilm. So they tested the agents on free-flowing microorganisms, and you add some antimicrobial agent and you kill all of them, no problem. And here with the biofilm, you kill all that are near the surface. But then the penetration is slow, and so those organisms that are hidden way within the biofilm, they may just get a stress situation, and they may even then, after some time, they only get small dosages. They become persisters, as it's called. They are resistant to the penicillin or whatever is used. And then after some time, they're already there, and then they can start growing and become dominating. So that's part of the explanation that has been used to explain why such agents have developed so fast. Another topic that some of my friends at the center was working on, they had started working on it when I was there, was to see that there seemed to be episodes where microorganisms from way inside the biofilm would all of a sudden just leave. And that was the first and probably so far the only article that has been published in Science on biofilm. Here, and what they call it quorum sensing, saying that the microorganisms way inside the biofilm, they are working together. So So the organisms then that are no food, life is rough, they decide, let's not stay in this biofilm anymore. How to get out of there? Well, sending out this quorum sensing signal to all the neighboring cells, And all of a sudden, there is enough of them that they just kind of dissolve the biofilm, and out comes all the microorganisms, and they leaving a big hole in the biofilm. That was a pretty big discovery, but it took many years to really document this. And what they saw was if they added or looked at the same type of concentration in the suspended culture, they saw no effect. So for this signal chemical to work, the organisms had to be close together, and that way they could respond as together. So since then, there's been lots of work going on trying to utilize this. Can you send a signal to a biofilm that it's time to leave? Extract this chemical, make lots of it, and apply it, maybe to the surface, somehow make them leave, but nobody has succeeded in that yet. The best case I've seen of microorganisms or control that is more, call it natural, is probably from Australia, where they've been putting together, or they've been extracting chemicals from seaweed. Because if you ever go swimming in the ocean fjord, you will see there is not biofilm on seaweed. So the idea is that seaweed has some way of keeping the microorganisms from colonizing. And they have been able to find such chemicals, and they've been able to extract it, make it, and turn it into a way to control biofilm. Not very well, maybe, but quite good. So that's, hopefully that can lead to something so that we don't have to use all these nasty chemicals to control the biofilm, but something more natural. Quorum sensing, people are still working on it. There's been millions and millions spent on this research to turn it into something practical, and nobody has succeeded yet. But maybe this idea of extracting antimicrobial or anti-biofilm chemicals from nature is the most promising. So I think that's a good place to stop this lecture on the fundamentals of biofilms, and then we'll talk a little bit more about biofilm processes in wastewater next.